Hi everyone, and welcome to another online lecture. This slide is very important, where we look at the crossroads of metabolism in regards to where pyruvate goes. So last online lecture, we talked about the conversion of pyruvate to lactate, which is essential in order to recycle NAD that's needed for glycolysis. This lecture, we're going to talk about the conversion of pyruvate into acetyl CoA and onward to the citric acid cycle. To begin, we need to recall the location of the citric acid cycle, and this is in the mitochondria of every cell. Specifically, the citric acid cycle happens in the mitochondrial matrix, which is indicated by this, it almost looks like a water-like material that's inside the mitochondria. That's known as the matrix. And so pyruvate actually has to get through the outer mitochondrial membrane and the inner mitochondrial membrane in order for it to be converted to acetyl CoA. So it's good that the outer mitochondrial membrane has porons, which are small holes, which allow pyruvate, because it is only a small molecule, it's 3 carbons, so it's not that big, it can freely diffuse based on concentration gradients and a little help from a hydrogen symport through the mitochondrial membrane, which we'll talk about on the next slide. On the previous slide, I talked about porons. And this allows pyruvate to freely enter from the cytoplasm through the outer mitochondrial membrane. But now we're at the inner mitochondrial membrane, and pyruvate cannot freely pass. It needs to have a transport protein and a carrier to help it get through this membrane. That pyruvate is polar, and the inner mitochondrial membrane is almost resistant to anything. And that's because we don't want things getting into the mitochondria and ruining the energy production of the cell. So as such, pyruvate needs a transport carrier. And so its transport carrier is a symport with hydrogen ions that we see pyruvate getting pumped in at the same time as a hydrogen proton is getting pumped in. And then based on concentration gradients, we see the movement of hydrogen protons as well as pyruvate in and out of the cell. And the hydrogen proton pump that's happening here is affiliated with the electron transport chain and the hydrogen proton pump that's happening. So essentially, pyruvate is using the same channel system as the protons are using in order to pump and create a hydrogen gradient across the inner mitochondrial membrane. So now we're going to talk about the pyruvate dehydrogenase reaction, which is really that link between glycolysis and the citric acid cycle. But technically, it's not part of either. Once pyruvate passes into the mitochondria, it undergoes a reaction in which it is converted into acetyl coenzyme A. This is a reaction that is technically neither a part of the reactions of glycolysis or of the citric acid cycle. So we'll consider it as a separate phase of glucose oxidation, even though it is only one reaction. The citric acid cycle is a cycle that goes around in a circle, and pyruvate is not the substrate that enters this cycle, which is why it technically is separate from it. This is a very important reaction, and there's a lot going on. The reactants of this reaction, or the substrates, is pyruvate, the 3-carbon molecule. We also have the NAD going into the reaction, and we have a large molecule, which is called a coenzyme A. And you can see this in the figure by it says HS, so hydrogen bound to a sulfur, combined with a CoA. The CoA itself is 21 carbons long. Okay, so we have pyruvate, NAD as the coenzyme, and then a huge coenzyme A molecule. So we really have almost 24 carbons going into this reaction. The product is acetyl CoA, which is a 23 carbon molecule because we're combining pyruvate, the 3 carbons of pyruvate, with the coenzyme, which is 21 carbons, makes 23 carbons because we're releasing a CO2 molecule, which is 1 carbon. We also have the product of NADH and a 1 carbon molecule known as CO2, carbon dioxide. The biochemical processes are dehydrogenation or oxidation, and because we are releasing CO2 or a carbon group, it's called decarboxylation. So the 2 types of enzymes that are required would be a dehydrogenase and a decarboxylase. The possible enzymes names for this one is pyruvate dehydrogenase. So a couple things about this very important reaction. When naming an enzyme that catalyzes more than one biochemical process, dehydrogenation will take precedence in regards to the naming, hence the reason why it's called pyruvate dehydrogenase. As well, you can see that one of the products is NADH. And since 2 pyruvate are created for each glucose, the actual yield is 2 NADH. So all of the products and all of the substrates are multiplied twice for 1 glucose molecule. So we have 2 pyruvate, 2 NAD, 2 coenzyme A's, 2 acetyl CoA produced, and 2 CO2, 2 NADH as products. So just be mindful of that, because when we go to tallying the total of this reaction, we need to multiply it by 2 for 1 glucose molecule. So now I want to pay some attention to acetyl CoA, which is the substrate that enters the citric acid cycle. It is really important that you understand the molecule structure of acetyl CoA. First, let's look at what an acetyl group is, which is the top left figure. It is a carrier of 2 carbon atoms, where you see the inflection point that double binds to an oxygen, that's a carbon, and then there's a carbon at the end adjacent to the R group. The coenzyme A part of the molecule is huge and has 21 carbons in it. You will see it abbreviated as CoA because it is so long. We see a ribose sugar involved, some sort of base, we see lots of phosphate groups. It's a large molecule with 21 carbons. So when the two get put together, the acetyl group and the coenzyme A, you take the R group off the acetyl group, which then still carries 2 carbons, and you take the hydrogen off the SH group from the coenzyme A. So the question is, how many carbons will acetyl CoA have? Well, the answer is we have 2 from the acetyl group combining with the 21 carbons from the coenzyme A, so 21 plus 2 makes 23 carbons. So acetyl CoA that enters the citric acid cycle has a total of 23 carbons. Next off, we're going to start digging into the citric acid cycle and looking at all the biochemical reactions involved in it.